Hello, General Biology. This is our transcription and translation video. There's a lot, so let's do it. Transcription is the process of copying DNA, a gene from DNA, into mRNA, and translation is a ribosome using that mRNA to construct a protein. But naturally, it's a little more complicated than that, so we've got to get to it. Let's start with transcription. We're going to start with transcription. Transcription is creating mRNA, and it occurs in the nucleus. What happens is a reader protein called RNA polymerase attaches to a gene at the start of the gene, attaches to the DNA at the start of a gene. And as it travels along, it separates the two DNA strands. And it does this over here by breaking the hydrogen bonds in the DNA so that the two strands, the hydrogen bonds are keeping the two strands kind of braided together and they separate out. Well, the nucleus is full of other nucleotides because nucleic acids are made of nucleotides. And these nucleotides are called free nucleotides, and they are attracted to the open nitrogenous bases of the DNA, just like these are. They each kind of find their place, just like the pairing that we already know. Guanine bonds with cytosine and adenine bonds with uracil and so on. So these free nucleotides have covalent bonds formed between their sugar and phosphate. So now we're making a new sugar or phosphate backbone, only instead of being a double helix, it's a single strand because we are making mRNA. When the mRNA is completed, it leaves the nucleus through transport proteins in the nucleus so that it can be in the cytoplasm where it can find a ribosome and we can start translation. Translation is the process of making proteins, and it starts when the ribosome clamps around and forms around the, let me start this over, it starts when the ribosome, whoa, come on now, reload. There we go. Starts when the ribosome forms around the mRNA, and then the mRNA is pulled through the ribosome one codon at a time. Now, a codon is 3 nitrogenous bases in the mRNA, and what happens is these 3 nitrogenous bases in the mRNA, it's kind of slow right now, but they attract tRNA, which are the blue things here. The tRNA each bring an amino acid. So the codons here attract the tRNA bring an amino acid, and the ribosome connects the amino acids together to make a polypeptide. Now what this animation doesn't show is that once they're completed, that polypeptide, which is unfolded, will fold into its protein shape. And of course proteins have different jobs like receptor, enzyme, transport, and structure proteins. So the summary of all of this, the major idea we want to get at, if I can click this, is that the order of nitrogenous bases in DNA determines the order of nitrogenous bases in RNA, which determines the amino acids in the protein, which will determine the shape of the protein, which will determine the traits or adaptations of the cell and the organism. So let's review. Transcription is the process of creating mRNA and occurs in the nucleus of the cell. The reader protein, called RNA polymerase, attaches to the DNA at the start of a gene. RNA polymerase moves down the DNA, uncoiling the two strands by breaking the hydrogen bonds in the base pair ladder. Free nucleotides are attracted to the open DNA nucleotides through hydrogen bonding. Once in place, covalent bonds form between the sugar and phosphates of the free nucleotides to make the mRNA, and when complete, the mRNA leaves the nucleus through transport proteins to find a ribosome. Translation is the process of making protein, and this occurs in the cytoplasm. A ribosome forms around the mRNA, and the mRNA moves through the ribosome one codon at a time. A codon is a set of 3 nitrogenous bases in the mRNA. The codons attract tRNA with hydrogen bonds. Each tRNA brings an amino acid. The ribosome takes the amino acid and covalently bonds them together to make a polypeptide, and the polypeptide folds into a protein when finished. So our final summary is that the order of nitrogenous bases in DNA determine the order of nitrogenous bases in RNA, which determine the order of amino acids in a polypeptide, which determines how a protein folds, which determines the adaptations of a cell or organism. I hope this helps. Struggle well.